Hello, my name is Mansfield, and my creature feature video is on the horseshoe crab. Below are the basic taxonomic levels of the horseshoe crab. It's in the Animalia kingdom. Its phylum is Arthropoda. The subphylum is Chelicerata. Its class is the Merostomata. Order is the Xyvocera. The suborder is the Xyvocerida. Its family is Limelidae, the species is Polyphemus, and the horseshoe crab's scientific name is Limulus polyphemus. Here we just have a brief history of the horseshoe crabs. These creatures are basically living fossils, and their real fossil relatives can be traced back to the Ordovician period. That's about 450 million years ago. And modern day horseshoe crabs can actually date back to the Jurassic period, which is about 200 million years ago. Horseshoe crabs tend to be found in coastal waters and reside along the soft sandy seafloor. When breeding, they're located in intertidal zones during spring high tides. Typically, horseshoe crabs grow between 14 inches to about 19 inches during their lifetime. So what is the actual feature of this creature? The horseshoe crab has a very unique trait about itself. This trait is that its blood contains a protein called lamellus ambisite lysate, or LAL for short. This protein is actually responsible for the shade of blue that a horseshoe crab's blood is. This blue actually stays intact when exposed to oxygen as well. And the reason this is such an interesting feature is that this blood and the protein within the blood are used by humans on the daily in the medical field. So what are the actual medical uses of LAL? Although the medical uses are fairly limited for the LAL, it's actually used every day, and it helps mankind in our ability to live healthily. Since the blood plays such an important role in modern day medicine, over 400,000 horseshoe crabs are bled annually by humans. So the protein in LAL actually has a pretty miraculous feature. It's used basically to test anything that may enter a person's body for contaminants. These things include IV drips, medical implants, and every single shot a person may get in their lifetime. So now that we know that the lamellus ambisite lysate is used in the medical field to kind of just track down contaminants, we can ask the question, how is it actually able to do this? In the case study by Miss Noyes, we actually learned that the blood is able to alert testers of toxins by coagulating around bacteria. And coagulation is actually just the process of changing a fluid to a solid or semi-solid state. It's kind of like blood clotting. So the blood of the crab actually does this process so effectively that it can coagulate around 1 part per trillion of invading bacteria. So just to put that into a little bit of perspective, that's equivalent almost to 1 grain of sand in a typical pool of water. The reaction also takes place in less than an hour in contrast to 2 or more days for mammal blood. So not only is this a very accurate way to find contaminants, it's also fairly quick. And since the LAL within the blood is the perfect substance for both of those reasons, it actually gets quite expensive. About 1 quart could actually sell for as much as $15,000 within the medical industry. Now that we know that the LAL is located within the blood of the horseshoe crabs, we can ask another question is how do we actually get this? And the extraction process, although at first it can seem kind of tough and daunting, it's actually said to be quite simple by people in the industry. Mainly it's simple because the animals will come right up on the shore by the hundreds and in better cases by the thousands during the mating seasons. This allows for the people in the industry to just go ahead and pick them up They inspect them. If they're injured, they're not going to keep them, but if they're healthy, then they'll be transported to the laboratories. While they're in these labs, they'll actually be hooked up to a specialized table that keeps them still. They're then injected to draw the blood by a big needle. And once that needle's in, they take about 30% of the blood as it moves to the heart. And then once they have enough blood and they've determined they no longer need the horseshoe crabs, they'll actually go ahead and drop them off in a new area other than where they were caught to make sure that they don't get caught again and lose enough blood to kill them. In theory, the process would eliminate any deaths because they take away the injured ones and they only extract blood from the healthy ones, but there's just a sad truth in that about 10 to 15% of those who are caught end up dying. And since there's only about 250,000 of these creatures left, 10 to 15% is actually a pretty significant percentage there. Future of LAL and horseshoe crabs in the field of medicine. As of now, the only true way to determine our medical equipment is 100% safe and contaminant-free is to use the blood of these fossils. However, the scientists and researchers within this field, they've been able to actually synthesize factor C within LAL, which is the clotting agent of this protein. Unfortunately, the synthetic known as RFC isn't approved by the FDA just yet, so this makes the wait for the synthesized version of LAL still in effect. We got a few years before the synthetic is available, so that means that within a few years we'll have this synthetic and horseshoe crabs, and this whole process of capturing them and extracting blood should actually be eliminated. With a synthesized coagulant, there would be no longer any need to capture horseshoe crabs, so this would actually increase their population a little bit each year because 10 to 15% of them do die off, as previously stated. So without this practice and need for horseshoe crabs, then that 10 to 15% would no longer be affected. So in conclusion, the blood and LAL found within the blood of a horseshoe crab is extremely important to mankind. It's important because the LAL and the protein within it, it's able to alert us of any toxins found within vaccines, IV drips, medical implants, and things along that line. Without this, we would actually be introducing these toxins directly to our bodies because we'd have no way of identifying them. Another thing to conclude is that factor C within the LAL is responsible for its coagulating properties. This is because factor C is a clotting agent. So what this means is that factor C is the property of LAL that makes it identifying and clotting around these toxins possible. Thirdly, we can conclude that it is an expensive process. It might be a little tedious, some would say unjust, but it is necessary in the field of medicine to make sure that everything is healthy and safe for human consumption. We can also conclude that the extraction process is simple and effective, but it slightly decreases the population. But this is actually looking like it's going to change because we have been successful with creating a synthetic of factor C. We just have to wait for all the approvals and the FDA to get involved. And then lastly, a synthetic factor C seems like a probable substitute for the blood in the near future, so this will eventually make the whole process of capturing these horseshoe crabs and extracting their blood obsolete. So then we no longer need to do it, and in turn it would just increase the population of these horseshoe crabs, as we said a little bit earlier. And here we just have the citations for the whole creature feature video.